just for a little, we should backtrack and just overview the immune system and kind of how it works, what T cells are. I know because you guys have explained them to me 3 times in the last week.
Sure.
um, but for those listening who have no idea what a T cell is, or let's kind of backtrack and just overview what the innate and the adaptive parts of it are, what the T cells are, and Tommy and Chris, you guys mentioned B cells before, and some different things. So, Chris, you want to kind of lay out the summary, I guess?
Sure. Yeah, I can go through it. So there's a couple different kind of, if you look at the broadest level, there's a couple different aspects of the immune system. So if we look at the top level, there's the innate immune system and the adaptive immune system. And the biggest difference is the innate system is hard coded. So if you're thinking of it from an example of programming, right? You can program a computer to look for specific inputs, right?
Mm-hmm.
That would be like if you do like a drop-down menu, right? So you can, if you're on any given website and there's a drop-down menu, say you're putting in your location, right? So you put your address, then you put your street, and then it has a drop-down menu for state, right? The program has a hard-coded set of 50 responses that it can accept. Alabama, Mississippi, Massachusetts, Boston, or not Boston, but any of the states, right? We consider ourselves a state, thank you. But so those are hard coded, right? The system doesn't know how to interact with anything that is not one of those 50 answers. So the immune system has a series of programs like that as well. So it knows to look for certain things, like things that tend to find themselves in the extracellular wall of bacteria, right? Lipopolysaccharide, or different variations on RNA and DNA, right? So that you'll look for cyclic DNA, which is not something we make and expose a whole lot of, or different types of RNA molecules that tend to be in pathogens, not in humans. So there's some very specific inputs that the immune system knows. Hey, if I see one of these 50 answers, something's up. But it doesn't know how to process something that's not one of those 50 answers, right?
Mm-hmm.
So in evolution, there's a little bit of an arms race going on with the immune system and everything else that's out there. So, bacteria doesn't just sit there and say, well, you got me, you've got my number. I always use lipopolysaccharide, so I guess you win, right? It's over. We're just going to go home. So that instead, what they'll start to do is the pathogens will start to use a number of mechanisms to hide these things. And so then you get this evolutionary back and forth where, we're looking for signal A, and so they hide signal A. And then we learn to look for signal B, and then they hide signal B. And so that's where the adaptive immune system comes in. So the adaptive immune system learns on the fly what it's looking for. So it's slower. The innate immune system knows, look, I'm looking for the 50 answers. It's encoded in the DNA. It's very quick. It knows exactly what it's looking for. The adaptive immune system has to watch the system develop and watch the infection and say, oh, hey, look, there's a target. There's a target. And so then it learns and develops a response to something in particular. And so that comes with positives and negatives, right? Like I said, the innate immune system's fast. It's quick. It's very powerful. It knows if it gets triggered, it should probably wipe out whatever that is. But if something doesn't fall into those 50 answers, it's basically inert. It can't find anything that falls outside of the box. The adaptive immune system is slower. It takes a while. It has to learn what you've been infected with and learn what it can target and how to respond. But it's super versatile. So in theory, your immune system can respond to things that don't exist today. So if you were to, generate some mystery molecule tomorrow and inject it, it's never existed on the planet except for in this lab one time, and you inject it into a passerby, they'll probably respond to it and generate a specific immune response to it because their adaptive immune response can adapt to the new environment.
Okay.
So that's the advantage of the adaptive system.
Cool.
Yeah. And just to follow up, so like one, of the challenges with using, immunotherapy to treat cancer is that, there's a lot of safeguards that Chris, you mentioned already. And a lot of those are to protect, the immune system from recognizing your own cells. And the issue with cancer is that, cancer arose from your own cells that acquired some set of mutations, and they may be a lot, it may be not that many, and so the immune system has to be able to recognize those very small differences, right? Like the majority of things are going to be the same as other cells in your body, so there's kind of a limited shots on goal. And so the kind of the goal of different types of immunotherapy are how can we get at those few shots that we have, right? And so there's lots of things that influence that, but yeah, we can get into that.
And that's what you're working on, right, Tom? The specific parts of what they should recognize?
Sort of.
Or all three of you, really, I guess?
Yeah, I think that's sort of everyone's end goal. At the end of the day, we do it a bunch of different ways, but we're all trying to enhance that shots on goal, like you said. Our objective is to allow the immune system to find that very specific molecule and go to town. And one of the important things to note that I think both Katie and Chris touched on and did a good job is the versatility and the flexibility of the adaptive immune system targeting system. So if you think about it, right, the objective of your immune system is to recognize things it can't predict, right, and be able to deal with it when it happens in real time. So if you want to have a T cell, for instance, that's capable of responding to a particular pathogen, there are kind of two basic ways you could go about encoding that. One, you could have a very specific segment of DNA that codes for every possible pathogen in the universe. Your DNA would be astronomically large. It's unreasonable. People have done the math and they've shown that the human genome is about 3 billion base pairs. And if you put every single one of those into just immune cells, you still wouldn't get the flexibility we have now. So teaching you by having everything base encoded is unreasonable. So what we actually do is you have a controlled system of randomness is how I'll describe it. And in short, in these T cells and in B cells, your genome actually selectively rearranges and changes. It is a controlled mutation, but it is a mutation. And the resulting feature is that you have T cells that are able to respond to random things. So if you stop there, you'd actually have a problem, right? Because you'd have a bunch of T cells that could be specific for you, right? Your proteins. So there's actually a training ground for the T cells called the thymus. And the short version is immature T cells go in, they rearrange to be able to be specific for a random thing, and then your body exposes that T cell to a bunch of what I'll call healthy, normal host proteins. And if the T cell reacts, it's killed. If it doesn't react, it's allowed into the periphery because it essentially is able to recognize something that isn't you, right? But it is a random process. We don't know what it is specific for. We just know it's not you. And then it goes out into the periphery. And if it encounters something that it is capable of engaging with, it will attack it. But this becomes kind of an interesting feature, right? Because you think about bacteria and viruses and parasites and things like that, and they are very much not you, right? Pretty much anything they have is considered foreign. So there's a good chance that one of your T cells will recognize it as not you and kill. The issue comes with cancer is it sort of is you, right? It's you, but it's not you. So it kind of walks this interesting new line of, as Jenna was saying, a bunch of the molecules will be you. So your T cells that would have theoretically recognized that molecule have been erased. So the question is, do you have some T cells that are capable of recognizing a mutated version of you? And that's what all of us are in some capacity working on is these T cells do exist. So how do we support them? Because there's not usually a lot of them. And they are attacking a version of you. So there's a lot of defense mechanisms in place to prevent that. So how do we selectively overwhelm those defense mechanisms and allow the T cell that is selective for a mutated version of your protein to do its job and kill the cancer without going off target and causing a bunch of autoimmunity issues?
Yeah, and like to follow up on that. So I think, relatively recently in terms of, science, like this, it was thought that the immune system couldn't be used to target cancer because of that, right? So, these like checkpoint inhibitors were kind of the first example of, that this could work, right? And actually works surprisingly well in a lot of patients. And so I think that, now there's a ton of interest, right, to try to find ways to make it better, or, alternative, pathways to get there.
So I think is one of those ways to, Tom, you mentioned turning off the inhibitors, I guess, that would not allow the T cells to do their job. Is that what the PD-1 does?
It is, yep. So there's a couple of different versions. So we call them checkpoint receptors. At least I think there's a couple of different versions of this name, but that's one of the common ones. And the idea is as everyone's talking about so far, that let's take it through a normal infection, right? Let's say you got a cut in your arm, a bacteria gets in and it's dangerous. That bacteria will initially be recognized by your innate immune system, which will do its best to handle it. It will try and phagocytose it. It will try and destroy it. If it can, that's where it ends. If it can't, it will try and hold the line. And one thing that we didn't talk about earlier is that the innate and the adaptive immune systems, while separate, are linked. So take, for instance, that the innate system has something called a macrophage. A macrophage will try and phagocytose bacteria and destroy them, but at the same time, some of them will head to your lymph nodes and will start to activate the adaptive immune system, essentially holding the line while we call for specific reinforcements, right? We really want your T cells to get in there. We're going to hold the line, but we need you to get here as soon as you can. So it's kind of this interesting system where everything plays nicely with each other.
Yeah, and I mean, if we get into vaccines and COVID-19 later, so I think one other important feature of the adaptive immune system is that it has a memory, right? So if you do have a T cell response against a pathogen or against cancer, so these cells will proliferate. You'll make a bunch of them, to eradicate their target. But then after the infection is cleared, you don't really need all of them around anymore. But your body wants to remember that pathogen in case it ever sees it again. So there's a specific type of T cell called memory T cells, and these aren't present in great numbers, but they're constantly kind of circulating and like on patrol for in case that pathogen ever comes back. So that's part of, along with B cells, which we can maybe get into a little bit more, that allows you to have a memory of an infection. And so if you have, some type of cold or something, or then you will have resistance to it going forward because now your immune system's been trained already, right? It doesn't have to go through all these steps of the maturation process and proliferating. It's kind of armed and ready to go.
I realize I don't think I fully answered your question, but Katie kind of jumped in and saved me. After you have this immune reaction, right, you want to generate your memory cells, and as she said, you don't really need all of those cells anymore. So this is where something like PD-1 would naturally come up and allow you to start shutting down that immune system to generate those memory cells. So you don't have an active immune response forever, just until the pathogen is cleared, then you make your memory cells. So that's where PD-1 kind of classically comes in.
Right, because after the cancer is gone, you wouldn't want your T cells to continue attacking your own cells. So once the drug is stopped, they would stop doing that.
Right. And that's sort of where Chris was talking about earlier is we're trying to release the brakes on the immune system. So they're being inappropriately stopped early. So we're going to temporarily intervene, prevent the cancer from turning off the immune system using this PD-1, PD-L1 axis. And then once the cancer is eradicated, we'll take you off the drug, your immune system will shut down, it'll make these memory cells, and it'll just sort of patrol and survey for the cancer should it ever return. That's the ideal ending for us.
That makes